Hello, my name is Danny now, and I'll spend a few minutes going over Punnett squares and how to apply them for solving typical biology problems that you might see in class or on a standardized exam. Now, what are Punnett squares? As you may correctly surmise, they were named after the creator, Professor Reginald Punnett from the UK. They are a visual way to show the genetics of mating between two parents. So with this visual tool, you can try to predict the possible genotypes and phenotypes of the offspring. An example of what a Punnett square looks like is shown here. It's typically a box with different squares in it, hence the name Punnett square. And the top row and the side rows are used to show the genotypes as well as the gametes, sperm or egg produced by the particular parent. So say for the top in the teal, we'll arbitrarily call that, that'll be parent one, we'll arbitrarily call that the father. The father happens to be heterozygous for a gene, that is, he has a dominant big G and a recessive little g for a particular gene. And I've represented here for clarity the father's alleles as letters in white. Because he produces sperm that are haploid, that is, they only have one allele of the gene, this box here represents one sperm, and it has a big G allele. The other sperm produced by the father has a little g. Sperm can only contain one allele, not both. Similarly, for the mother, for the eggs she produces, they can either contain a big G or a little g, but not both. The offspring is shown in the green boxes in the middle here. So for example, in this particular offspring, has a big G from the father, shown in the white, and a big G from the mother, shown in the black. So this particular child is homozygous and has both dominant alleles for the particular trait. In contrast, this child here has only little g's or recessive alleles for this gene, and their remaining children are heterozygous. Now, how do you use Punnett squares? And I guess one way you oftentimes see them is if you ask a question about what percent of offspring would have a given trait. For example, a phenotype of eye color, when you're told the phenotype and genotypes of, and potential genotypes of both parents, given whether a trait is dominant or recessive. So take, for example, coat color in animals. It could be determined by a single gene with two possible phenotypes. We'll call it a dark coat and a light coat are two phenotype possibilities. Allele C is the dominant allele, and that is responsible for a dark coat. Little c is a recessive one that produces a light coat. So the question is, if you have a male with a dark coat who mates with a female with a light coat, is it possible to have any offspring with light coats? And here there's actually two possibilities. Again, we let the top box represent the father's sperm. The father has a dark coat, so we know he's got at least one big C. He could have a second big C, or he could have, since the trait of big C is dominant to little c, he could have a recessive allele. We don't know that, but let's set the crosses. And we know the mother has a light coat, so she has to be homozygous, little c, little c. You go through the crosses, an example here, if the father only has big C alleles, all the offspring are going to be heterozygous big C, little c, and so since big C is dominant to little c, all the offspring will have dark coat. This possibility here, because the father has one recessive little c allele, you're going to have a mixture. You're going to have half the kids will be big C, little c, hence they'll have a dark coat. The other half will be little c, and they'll have light coat. So in this case here, you have half the offspring with a dark coat and half with a light coat. Let me go into another example that seems to be a favorite for test makers, and this is where the questions about sex-linked disorders comes into play. Now, sex-linked disorders are genetic diseases where the gene responsible is actually located on one of two sex chromosomes. A classic textbook example would be a question about color blindness in humans. This is due to gene found chromosome. On the X chromosome, it is recessive, so that if women only have one copy of the mutant allele, they'll actually have normal vision, since women have two X chromosomes. However, a male, which only has one X chromosome, the other being a Y, if they inherit just one mutant allele, they will have color blindness, even though the trait is technically recessive. So in this example, let's say we represent the mutant allele as X star for color blindness and X is wild type. The question is, does a colorblind male, if he mates with a non-colorblind female, will all male offspring also be colorblind? And look at the possibilities here. Let's represent the father again, shown in teal. You know that he's colorblind, so he has to have an X star allele. He's a male, so he's got a Y. That's the only possibility for him. The female, the mother, could have true normal X's, or she could be a carrier with one X star. In either case, her phenotype would be normal vision. When you do the crosses, scenario 1, you're going to get the situation here. You're going to get these two offspring here will have one mutant allele, but because of the wild type one, they will have normal vision. In the males, since they only inherit the X chromosome from their mother, they will be genotypically normal and will not be colorblind. So all males will have normal vision. However, if the mother is a carrier, and you just look at the males, you're going to see now that one of the male offspring will be colorblind with the X star, the other one will have normal vision. So half the males will be colorblind. And as a side note, if you look at the females, you're actually going to have one female who inherits two colorblindness alleles, and she will actually be colorblind as well. Her sister will have normal vision. So in summary, Punnett squares are a visual tool to help you solve genotype-phenotype problems that you'll typically see. And the problems may involve sex chromosome and genes, with a classic example being color blindness in humans. Now keep in mind, for this method to work, the genotypes have to be represented by clear phenotypes. For example, if questions about coat color and there are alleles big A and little a, the resulting possible genotypes are big A, little a, big A, or little a. You need to have distinct enough coat colors to tell the different genotypes apart. If you have, say, variable penetrance, where the same genotype could be showing up differently in different individuals. For example, big A, little a, if it shows up in black in one individual and silver in another individual, then the Punnett square approach unfortunately won't get you the answer you want. If you'd like to learn more, feel free to reach out to me. I thank you for your attention and time, and I look forward to working with you in the future. Thank you.